Are we ready? You look ready. I have not met our next speaker, Laura Alvarez, but what I've learned is that Laura is a very highly experienced science consultant to CrueltyFree International, and where she gets a chance to use her background in biomedical research and clinical microbiology. And I think this is such an important concept here to secure the necessary evidence-based animal research alternatives. So, Laura.
Hi, everyone. I hope you enjoyed your lunch. Some of you are still enjoying your lunch. Thank you so much for that kind introduction, and I'm really happy to be here. Unfortunately, Katie couldn't be here today, so I'm going to have to replace her. I hope I do as good a job as she would. So today I'll be providing an overview of recent developments in alternatives to animal testing. Space. Oh, there you go, perfect. All right, so first off, I will explain a little bit about who we are and provide some brief background on our organization. I will then go through some statistics. Even though we've heard a couple of different statistics today, I'll give you a few more, just to give you an idea on the scale of animal use across the world. The focus of the talk will then be on alternatives to animal experiments, starting with recent progress in the use and development of these methods, and then moving on to their implementation and some of the main barriers to replacing animals. I'll then finish up with some targets for change. So first of all, CrueltyFree International is the leading organization working solely to end animal testing worldwide. We do this through award-winning campaigning, political lobbying, undercover investigations, scientific and legal expertise. We also promote the use of cruelty-free products through our Leaping Bunny program. We work with governments, regulators, companies, and partner organizations worldwide. We are also members of the European Coalition to End Animal Experiments, which is an alliance of 24 animal protection organizations across Europe. It was founded in 1990. So our work under the coalition includes ensuring that the EU cosmetics ban is upheld and also providing scientific, legislative, and political expertise to improve EU legislation and reduce animal experiments. Both the Coalition and CrueltyFree International are also part of ICAPO, so it's the International Council on Animal Protection and OECD programs. ICAPO works to incorporate alternatives in OECD activities, so in chemicals testing, and we're also members of ICAPO's sister organization, which is called ICAP, so the International Council on Animal Protection and Pharmaceutical Programs, which works to promote animal protection in international pharmaceutical guidelines created by the ICH for human medicines or the VICH for veterinary medicines. So just a bit of history. We are almost 120 years old, and we were originally established in 1898 as the British Union for the Abolition of Vivisection. We changed our name to CrueltyFree International in 2015 to reflect the work that we do globally. So the picture on the top right is just to show you one of our public demonstrations outside the UK Parliament, where we almost had a bill passed to end testing in dogs. So these pictures are just to demonstrate the level of feeling around animal experimentation even all of those years ago. So to give you some statistics, our most recent estimate is from 2005. So Dr. Katie Taylor and colleagues estimated that over 115 million animals are used worldwide each year. Although 79% of countries didn't actually appear to publish statistics on the use of animals, we used an estimation model based on the countries that do publish those statistics to arrive at the estimate. As you can see, the top 5 users were the USA, Japan, China, Australia, and France. We are actually almost finished with a new estimate for 2015. Spoiler alert, the number's gone up, but we hope to publish this early in the new year, so watch this space. So I'll start with the US, excuse me, since they're currently the biggest users in the world. So according to the most recent report, in 2017, US laboratories reported that almost 800,000 Animal Welfare Act and regulated animals were used in experiments. As was mentioned earlier, this number doesn't actually include the majority of animals that are really used in experiments, as mice, rats, birds, and other species are not counted or covered by the AWA. So the statistics show that a total of 62,055 animals were used in Category E experiments in which no pain relief was provided. So this includes quite a large number of monkeys, pigs, and dogs. And while there was an overall reduction of 3% in animal use since 2016, the number of dogs and monkeys has actually increased. So almost 65,000 dogs were used in the US alone, and almost 76,000 monkeys were used. So this represents increases of 6% for dogs and 7% for monkeys since the previous year. Now moving on to Europe, this chart is just to show you that the number of animals used in Europe is not really going down. It's actually been around 11 to 12 million for a long time now. The latest official statistical report we have from the EU Commission was published in 2011. Before that, it was published every 3 years. However, with the introduction of the new directive on animal experiments in the EU, the Commission's been given until the end of 2019 to publish the new collated statistical report in the newest format. So instead of waiting until 2019, we've decided to produce our own report on the use of animals across Europe in 2016. This one's also going to be published probably in the new year. So basically, we collated all of the national statistical reports from all the EU member states that are published on the Commission's website, and we found that 10.5 million experiments were conducted on animals across the EU in 2016 alone. So this represents an overall increase of 1.4% from 2015 to 2016. Germany, the UK, and France were the top 3 users, and Ireland, the Netherlands, and Czech Republic were the countries that reported the highest levels of suffering in animal experiments. So over 23,000 dog experiments were conducted, with UK, France, and Germany being the top 3 users, and almost 10,500 monkey experiments were conducted, 94% of which involved macaques. France and Germany were the only 2 countries in Europe to test on baboons. And once again, the UK, France, and Germany were the top 3 users of those species in that order. So this is just a pie chart just to show you the overall purpose of experiments in Europe in 2016. So I think it was mentioned earlier that the majority of animals are used for basic research purposes, which includes curiosity-driven research, research designed to answer scientific questions that researchers might speculate might be useful medically in the future. It's normally carried out by universities. 25% of testing in Europe is for regulatory testing, so this is standardized tests designed to see if medicines and other products are safe and effective. And then 21% of experiments are for translational and applied research, which covers non-regulatory testing of medicines and other products. So as you can see from this chart, the breeding proportion actually looks like as you can see from the chart, 4% of animals were used to breed GM animals. So these animals are not used in those experiments. It's animals used additional to the experiments that are conducted there. It might sound like a small percentage, and it is because the directive only requires that EU member states report the number of animals used to breed GM animals with a harmful defect. That's quite a mouthful, but basically it excludes all animals used to breed GM animals without a harmful defect, and also all normal animals that are used in the breeding process for genetic modification. So there's actually a huge number of animals that are missing from here. This chart also doesn't show the use of actual GM animals used in experiments because that will fit into each of those different categories. I hope that made sense. So here's a list just to show you the level of regulatory tests that are still being conducted in the EU for which there are available alternatives. The numbers are actually likely to be a lot higher than what's written here because even in the 2016 reports, 7 member states are missing figures on regulatory testing, so including some big ones like France, Netherlands, and Italy. We're also missing data from Portugal, who are the only member state to have not published a statistical report since 2014. So slap on the wrist from Portugal. I'm actually Portuguese. As you can see from the test, there's still quite a large number of tests that have validated alternatives that are being done. So we have skin irritation and corrosion tests. I'll tell you a little bit more about the alternatives to these in a minute. We have eye irritation tests still happening, skin sensitization, pyrogenicity, and then we included batch potency testing here because we believe that a large proportion of these tests are actually for testing Botox and for vaccines, of which there are alternatives. So now just to give you a little bit of information on Jared's chapter, he's our senior research scientist, Dr. Jared Bailey. He's also written a chapter in this book, chapter 19, Genetic Modification of Animals, Scientific and Ethical Issues. I'll just touch a little bit on it because it kind of ties in with the statistics. So the sheer scale of GM animal use is hard to determine. Many animals are bred and not used, and not all of them are recorded in the statistics, like I just mentioned. But we do believe that the use of GM animals is a significant cause of yearly increases in the number of animals used in experiments around the world. So just to say that the UK is actually the only country that reports and records all animals used in GM breeding process. So they do also record the number of normal animals and the number of animals that are used to breed GM animals without a harmful defect. So this is kind of just helps you to see the scale of probably actual GM use. So basically in the UK in 2017, half of all experiments, so 2 million experiments carried out, involved breeding of GM animals that were not used in further experiments. And 38% of actual experiments used GM animals. So I won't go into all the welfare concerns for these, but they are considerable, and suffering has been acknowledged at every step of the GM process. Now here's the buzzword. Everybody's heard about CRISPR. It's in all the news articles at the moment. So it's basically an inexpensive, relatively simple, and a quick way of producing GM animals. Many are claiming that it's more accurate than traditional methods and that it will help reduce animal numbers because it's so accurate. In reality, though, CRISPR is only likely to make the problem associated with GM production worse by simply increasing the number of animals that are created just because it's easier and faster to do so. We believe that this will more than offset any potential benefits. While CRISPR may be more efficient and specific at generating desired genetic modifications, it's far from good enough. Studies have shown that the efficiency of CRISPR to insert a particular gene in an animal is less than 4%, and to delete a particular gene in an animal is around 7%. CRISPR also introduces unwanted or off-target mutations in addition to those that are intended that may have serious consequences. Perhaps the most troubling thing is the increasing use of CRISPR in larger animals. So some scientists are calling for more GM monkeys and dogs to be created in an attempt to fix rodent models that are increasingly failing. Just last month, for example, 2 experiments that involve the use of CRISPR in monkeys and dogs were published in high-profile scientific journals, Nature and Science. We actually wrote a letter to the editor of Nature to complain about the severity of the monkey experiment in particular and the lack of consideration for animal welfare in the study report. Just very briefly, scientists in China used CRISPR to knock out a gene in monkeys that they believed was related to longevity and the aging process. Sadly, out of all the babies that actually made it to full term, all of them died shortly after from severe retarded development that resembled a 2 to 4 month old fetus. Disturbingly, the authors concluded by saying that they're going to keep producing these animals as models for perinatal lethality syndrome in humans. So just to conclude with a nice point that Jared made, animals have always been bad models for humans due to species differences, and no amount of genetic modification can remedy that, even if those techniques were perfect, which they're clearly not. So instead of trying to improve GM processes, we should be working towards the greater adoption of more human-relevant technologies. So here's just a couple of recent opinion polls. I won't go into them in huge detail. They've already been mentioned, but just to show that the public support for the use of animals is dropping, and it's always conditional in that it works and that it's used for medicines. Once you start asking questions about specific animals or pain, then the support drops even further. So there was a poll in the UK in 2016 where only 18% of people agreed that it's acceptable to use dogs for medical research. There was also a poll in the US that found that only 47% of adults favor the use of animals in research compared to 89% of scientists. So I'll just leave, I won't go through all of them. So here are some key EU policy developments that have really set an example for change for the rest of the world. The most obvious one is the cosmetics regulation, which now banned the testing and import of tested cosmetic products in the EU. This in turn encouraged the development of alternatives to the traditional animal test. The EU chemicals testing legislation, REACH, was introduced in 2007 to assess the safety of 30,000 existing chemicals across the EU. This legislation highlighted the concept of animal testing as a last resort, which also encouraged the development of alternatives. And finally, the directive, which we've mentioned several times already today, on animal testing, implemented better reporting requirements to ensure that member states are more accurately and more regularly reporting the number of animals that are used in experiments. Also, just to point out, there's also a legal obligation in Europe to use alternatives if they exist, but this is a principle that's been in place in Europe since 1986. So this is just a little map just to show that the EU cosmetics ban is becoming a global trend, thankfully. So the countries highlighted in pink are where bans are already in place. So apart from Europe, there's also Turkey, New Zealand, Guatemala. And then in orange are the countries where bans are in transition, so Australia, South Korea, and Taiwan. And in blue, we have us, the US and Brazil and Canada, where bans are under consideration. And then the lighter pink areas, so China and Argentina, Russia, are where some other progress is being made. So we hit an important milestone recently. On October 4th, CrueltyFree International and The Body Shop took 8.3 million signatures to the United Nations to call for a global end to cosmetics testing in animals. So the petition calls on United Nations countries to create a global framework to end cosmetic animal testing internationally and permanently. Now, to move away from cosmetics, there's also been recent progress in the development of national 3R strategies. Most of these actually come from the US, which is very encouraging. So we have the National Research Council's Tox Testing in the 21st Century. That's quite a famous one. Then more recently, we have ICBAM's Strategic Roadmap for establishing new approaches to evaluate the safety of chemicals and medical products. NAMS is the buzzword, New Approach Methodologies. Then FDA's effort, which is still being drafted, FDA's Predictive Toxicology Roadmap. And then the EPA have their own strategic plan to promote the development and implementation of alternative methods within the TSCA program. And then on the bottom, I've just slotted in the Netherlands strategy, which is their transition to non-animal research. They are actually planning to phase out the use of animals in toxicity testing completely by 2025. So progress is being made. Now here's a list of some alternative tests that are already accepted for standard toxicity testing for chemical safety. So for skin absorption, the traditional test was a rat test where the substance was rubbed onto the skin of rats. Now we have ex vivo skin-based tests, that measure the amount of substance that passes through excised skin. So this was accepted by the OECD in 2004 as a standalone replacement. And then we have skin irritation and corrosion, which is a huge one where we've made a lot of progress over the years. So the notorious Dre's test, where the substance is rubbed onto the shaped skin of a rabbit, has now been replaced by reconstituted in vitro human skin models. So these models measure the extent of cell death in the presence of a substance, and they're made from donated human skin, excess skin from surgeries and things like that. These methods have also been accepted, and a testing strategy on how to use them in combination was adopted by the OECD in 2014. So for eye irritation, we have two different methods that are used. First, we have the excised eyes from hens and cattle that are killed for food, so the ex vivo models. These were adopted in 2009, and then we have human corneal epithelial models, HCE models, that are based on excised skin or corneas that were adopted in 2015. So the ex vivo models are used to detect non-irritants and severe irritants, and the HCE models can measure the extent of cell death in the presence of the substance to detect non-irritants. And finally, skin sensitization, that's another one that's made a lot of progress in recent years. I know there's a lot happening with that in the US right now as well. So the animal test, there's a guinea pig test and a mouse test, the LLNA. This can now be replaced by several tests that cover the adverse outcome pathway or the different steps that lead to skin allergy. So we have the DPRA, or the direct peptide reactivity assay, that measures the binding of the substance to proteins. That's the in chemico replacement. And then we have the in vitro keratinocyte and the HCLAT assays, which are both based on human skin cells, and they measure part of the immune response. So these methods can be used in testing strategies to replace skin sensitization animal tests. However, the strategies are yet to be formally accepted. So this is where it gets complicated. This is to show you the path of an alternative method. Unfortunately, the replacement of an animal test is a laborious and bureaucratic process. Even more unfortunate is that the process often has to be repeated for each sector of use. So just because you have an alternative that's been validated and accepted for cosmetics doesn't mean it can be used for chemicals, and that doesn't mean it can be used for pharmaceuticals. So the first step is development. So this is the stage in which the alternative is created, optimized, and initially tested. Academia plays a large role at this stage. Alternative centers such as the UK National Centre for the 3Rs, so the NC3Rs, and the alternatives charities are vital for funding this kind of work. Then we have validation. So this is where the method is independently assessed to ensure that it's reliable and accurate. This step is vital if the method's to actually progress to acceptance. There are internationally agreed principles for the way a method should be validated, but they're vague and they're not well understood. The key requirements include showing that the method produces the same results when tested at different times in the same lab, and also when used by other naive labs. And the results also need to be consistent, so the test has to do what it's designed to do. This process is laborious. It requires collaboration between several labs, and it also can be really expensive. And then we have the formal test method stage. So once there's sufficient evidence that an alternative method is valid, the next stage is to write up how the method should be performed as a formal test method. Negotiating how to conduct the method is often combined with further analysis of the validity of the method, and again, this process alone can take another several years. Then we have regulatory acceptance. This step does not automatically happen following the publication of a test method. Typically, several regional regulatory agencies have to assess independently whether the method can be used for their sector. Unfortunately, there is no official mandate either for them to do this, and they may need political pressure to act. Regulators typically accept methods by updating their guidelines, but they often in reality, only do this when a corresponding specific legislation has changed. And then finally, we have the deletion of the animal tests. So this is when sector-specific legislation is changed to replace any requirement of the animal test with the alternative. This process can take several years. Political pressure, again, is usually needed to instigate the deletion of the animal test, often following pressure from animal protection organizations. So as you can see, the first two steps are primarily science-driven processes. These can be sped up through appropriate funding and coordination, and the last three steps are regulation-dependent, and they can be accelerated by political will and regulatory enforcement. So just to show you an example of this process, we've included the timeline of implementation of the reconstituted human epidermis models for skin irritation and corrosion. Although the timeline might look relatively quick, you have to remember that this is just at EU level, so it's not even taking into account other regions. So first off, quite a few years passed between when the method was developed and when it was formally validated by ECVAM. It also took much longer to validate the method for irritation than it did for corrosion, even though the method is essentially the same. And then, as you can see, the steps don't necessarily happen in the right order, which can be confusing. So in this case, regulatory acceptance actually predated the formal test method. And finally, the deletion of the animal test for REACH came nearly 10 years after validation for irritation and nearly 20 years after validation for corrosion. And it still hasn't been deleted at OECD level. In fact, no OECD animal test has been deleted ever been deleted because of replacement. And here's another graphic just to demonstrate the complexity of the situation. Even though an alternative might be accepted for cosmetics, as I mentioned earlier, it might not also be accepted for pharmaceuticals or chemicals. On top of that, the method might be accepted at national level, but you still have to gain accepted at regional level and international level. There's also a lot of overlap, which can lead to even more confusion. So, for example, a cosmetic product can also be a chemical or a pharmaceutical. And the arrows are on the outside of the graphic represent all of the different people that can have a say or can influence the process. And unfortunately, many of these people are not actually even communicating or connected in any way with each other, just to further complicate matters. And finally, this is the last one I'll show you. These charts show the number of skin irritation tests and pyrogen tests that were conducted on animals in the UK between 2004 and 2016, even after the alternatives were validated. I already mentioned the skin irritation alternatives a few times, but I'll just quickly say that the pyrogen test in rabbits, which is used to determine whether a substance causes a fever response, can now be replaced by the monocyte activation test, which measures the fever response in human blood cells. So anyway, the point here is that animal testing doesn't end at the point of international validation. As you can see, it does slow down gradually, but there are inexplicable rises. And so there's, especially for the irritation, there's quite a few like big dips up there. So the reasons for this continued testing could be testing for other regions is quite a common reason. Also, scientific issue with the alternative or lack of availability or awareness that it exists. Also, need for product-specific validation, and a big one is lack of enforcement. So what is the future of alternatives? So rather than criticizing the ethics of animal tests, it's also important to focus on better science and set a future vision for toxicity testing. A paradigm shift is needed to move away from black box animal models and towards mapping human reactions at a more molecular and cellular level through the use of new technologies. So here's a few examples. First off, we have the concept of adverse outcome pathways, which I already mentioned for skin sensitization. These provide the actual biological explanation for a single toxic event. So again, the AOP for skin sensitization has been described, and it's made up of 4 key steps. 3 of these steps now have approved OECD in chemico and in vitro tests for. And then I'm sure you've all heard of organ chips. Human cells have been used to create innovative little devices called organs on chips. These can be used to study biological disease processes as well as drug metabolism. The end game is to create a human on a chip where different little chips are linked together to create one complex model that we could eventually use for drug testing. Rapid advances in technology have also allowed for the development of new techniques to safely study human volunteers. So we have brain imaging machines that can see inside the brain that can be used to monitor the progression and treatment of brain disease. And then microdosing, which can be used in volunteers to measure how very small doses of potential new drugs will behave in the human body. So what are the barriers to the implementation of alternatives? A major stumbling block when it comes to replacing animals is the current way that hypotheses are tested in science. The process is currently one of testing models of increasing complexity with growing confidence in the hypothesis as it successfully passes each hurdle. The most common justification for using animals is the apparent need to test a substance or an idea in a complex whole being before there's enough confidence that it can be safely tested in humans. So people have a hard time getting their head around replacing animal tests with in vitro tests because they're seen as more simplistic. And while it's true they are more simplistic in the sense that they only look at certain aspects of the process, but if they're testing the right aspects, like in the AOP pathways, then that should be enough. So what are some of the other barriers? Alternatives are currently validated against animal tests, and they're expected to produce equivalent results. So you won't always get a like-for-like replacement. We also have to remember that many of these animal tests that are being used were never themselves formally validated. Another big problem is the lack of international harmonization. So, for example, the rabbit skin irritation test is still being done in the EU, as I showed you in the numbers, for non-EU regulators, even though the alternatives are accepted in the EU. There are also bureaucratic delays between validation and regulatory acceptance. So, for example, on average in Europe, there are currently 4-year delays between validation and a method being included in the EU regulation. Another barrier is funding. Central and national funding of alternatives is very low, and it's ad hoc. So we conducted a survey in 2013, and we found that direct funding of alternative methods totaled only $18.7 million. That might sound like a lot, but that was actually only 0.04% of the total budget. The European Commission also funds alternatives, but again, as part of their total science budget, it only makes up 0.4%. Another huge problem is entrenchment in the scientific establishment. So new ideas that threaten the status quo can struggle to gain support. People are comfortable with doing what's always been done, and I experienced this in my career as a lab scientist. Another one that's come up quite often in recent meetings is the fear that alternative methods will not be accepted by regulators. So even though the methods are used, people are afraid to use them because even though the methods are out there, people are afraid to use them because they don't know if the regulators will actually accept them. And then finally, lack of enforcement. So for example, in the EU, it is illegal to do an animal test if an accepted alternative exists, but the tests are still being done. So that's a problem with enforcement. So how can we overcome these barriers? So first, we need to embrace and encourage a greater understanding and uptake of the adverse outcome pathway approach. We need to use a combination of methods, and we need to use more complex and sophisticated methods. So things like the computer chips, like the organ chips, also computer models, 3D cultures, organoids, the miniature brains that were mentioned earlier. All that type of new technology needs to be embraced. Also, another important one is those that are involved in the validation of alternative methods need to appreciate the regulatory use of the method and validate it accordingly. So for example, the alternative for skin irritation was originally validated to give a yes or no answer when chemical regulators actually needed it to determine whether a chemical was an irritant, a non-irritant, or a severe irritant. So they had to basically revalidate it to make sure that it could do what it's supposed to do. Another key one is working together to achieve harmonization of testing requirements. This is both across sectors and across countries. We also believe that rewards should be provided for the development and evaluation of alternative methods. There needs to be an incentive for people to do these things. We could encourage preferential funding for scientists who actually want to use alternative methods, and also make their funding applications open to public scrutiny. And then finally, we want regulatory bodies to be clear about what methods are accepted and to take responsibility in ensuring that they're used. So tying on from that last point, this is a concept that we came up with a few years ago for regulatory authorities. This is the ADAPT principle, so it's 5 steps that regulatory authorities need to take to make sure they're not blocking alternatives. So the first one is assessment. Who assesses if the method is appropriate for that sector? So does every regulatory body have a proactive mandate to identify and assess suitability of new methods for their sector? Then we have decision. When is a decision made on acceptability? Who has the responsibility for deciding whether an alternative method is suitable, and do they have a mandate to update their guidance and legislation proactively? Then acceptance. Where is the decision published? So have all regulatory stages been identified for each sector, and is there a body in charge of monitoring the progress of methods through these stages and accelerating them if need be? Policing, so why might animal tests still be done? Do all relevant regulatory bodies monitor the use of alternatives, and will action be taken if animal tests are done unnecessarily? And then finally, transparency. What is done to educate potential users? So does the regulatory authority have a clear and appropriate mechanism for informing all stakeholders of their decisions at each stage? So I won't go into huge detail on this slide. It's not in the chapter. I just threw it in here because it's another campaign that we've been doing. It's called our rat list. So basically, we created this list to highlight 10 of the worst animal tests, which in our view are either redundant or have valid replacements. So we believe that if we can remove these 10 tests alone, it could help save 500,000 animals in Europe. So you've already seen a few of these, and there's a couple of new ones in there. But for more information, this whole campaign's on our website. So now for some targets for change. So to continue the momentum from the EU cosmetics ban, I know not everybody likes the word ban, but there have been recent calls for more bans on things like testing household products and for testing on dogs and monkeys. And where this isn't possible, so where alternatives are not yet available to do these things, increased investments in technology should be made to at least allow targets to be set. So for example, we could set a target of reduction of 50% in national experiments by 2025. So that means even though the alternatives aren't available now, if we invest in the right technologies by 2025, they may well be. Targets are really important in other sectors, like in climate change and fishing, for example. It just focuses everyone in on the issue. We're not saying that fishing is bad, but you're sending the message that people need to think about it and it needs to be done sustainably. So these targets would feed into ethical review committees who would then have to make the right decisions. They would have to reject some applications, and it would also seep into the mindset of scientists who would need to think carefully before they submit those applications for new animal experiments. These targets would also increase the political will to fund alternatives and put in place necessary governmental and institutional schemes to develop, promote, and implement alternatives. And that's all. Thank you for listening.
Thank you. That was a great talk. Thanks for that. Any questions for Laura? Or was that too Europe-centered? I hope not. That was great.
It was a little Europe.
Thanks very much, Laura. That was really, really interesting.
Thank you.
This is just a quick question clarification request. When you said 0.04% of the research budget went toward alternatives, is that toward the development of alternatives or even alternative
It's all three
research
R's. It's actually 3 R's related, so it's not just specifically non-animal alternatives. So it's basically, the development of 3 R's methods.
So that wasn't that, like if I was doing a clinical test or an epidemiological test, you're not talking about research?
No, I'm talking
Okay.
about development of
Got it.
alternatives. Yeah, to actually funding new ways of doing things.
Great analysis.
Thank you.
Any other questions?
I don't know that I need the mic. I'm just curious of the countries that are making the most progress on cosmetics, when you put kind of the maybe column, where do you have the most optimism?
Well, things are progressing here quite well, I think. Where our campaign manager, Monica, is actually doing quite a lot of work to get bills passed, especially in California. There's a lot of progress being made. Unfortunately, we have the whole issue of state-by-state legislation, so I'm going to say there's a lot of progress being made here. Also in South Korea, there's talks going on over there. But really, obviously, the most progress is still in Europe. Other countries are still thinking about it, teetering on the edge, but we don't have any solid bans in place yet. Thank you. Thank you, everyone.
Thank you. Okay, I'm happy to introduce our next speaker, Lisa Kramer. She came all the way from Toronto, and she's a professor for finance, and she has been a visiting scholar in the Department of Psychology at Stanford University, and she was in California for a while. That's actually when I met her. We met in 2016 at the symposium on multidisciplinary research in effective animal advocacy in at Princeton University. And that was actually when I talked to her a lot, and then she actually contributed 2 chapters to this book together with Ray Greig, and I'm very happy that she's here and will present focus on one, but she will also say a little bit about the other one. So thank you. And
Thank you. Hey, thanks everybody. It's really wonderful to be here and to see many familiar faces and many new faces. I'm really grateful for the opportunity, not just to contribute the chapters to the book, but just to be here today. So thank you. So I'm here to talk about a couple of chapters. So the scientific problems with using non-human animals to predict human response to drugs and disease, plus a proposed debate format where these issues could be discussed amongst various experts. And this work is jointly conducted with Dr. Ray Greig, who founded and is president of Americans for Medical Advancement. And Ray is sorry he couldn't be here. Actually, it's a sunny day in Santa Barbara, which is where he lives, and so maybe he's not that sorry he's not here, but let's just say he's sorry he couldn't join us. So I'll start by talking about the second of the 2 chapters, which is looking at the scientific problems of using non-human animals to predict human response to drugs and disease. And so we're sort of starting with the realization that, practically speaking, based on observed data, non-human animal models are not good ways of predicting how humans will respond. And I'll make the bold statement that you'd actually be better off flipping a coin in terms of predicting whether a particular drug would be safe and effective in humans based on what you learn from looking at the animal model. And in fact, you would need much higher predictive value than even 50% or 60%, even drugs that have what seem like very high predictive values based on the animal models, even as high as 99% or higher, have been quite catastrophic in terms of the consequence for human response. And there are many examples that I will touch on in a little bit. And so a large part of what Ray and I talk about in the chapter is based on observed data, but we also talk about the theoretical case. So just even putting the data aside, we should know just from basic scientific arguments that animal models ought not to be expected to be predictive of human outcomes. And I'll walk through that logic as well. And finally, even if there were none of these great better methods that we've been hearing about today, even if those didn't exist, we would never want to use something worse than a coin flip to predict human responses. So, even if we weren't in the fortunate position to have all these alternatives at our disposal, we still would be on very shaky ground in continuing with the status quo. But fortunately, we do have lots of effective methods. They're available, they're improving all the time, and we need to continue to advocate for their adoption and find ways to encourage that to happen. So this graphic on the left of a mouse, first of all, I stole this concept from a presentation by Professor Jessica Bolker at a conference I was at a couple of weeks ago. And the graphic on the left came from a Nature article of hers that is called "There's More to Life Than Rats and Flies," making the argument that we do so much scientific research on non-human species and expect the results to apply, to generalize to humans, and it's just without any merit. Evolution in small changes in genes over long periods of time have led eventually to very different species. It's been tens of millions of years since humans and mice have shared a common direct ancestor, and even for humans and a closer relative, temporally and genetically speaking, chimpanzees, differences in our complex biological systems have led to nonlinear differences in the way our bodies overall respond to stimuli like drugs and disease. And so, this expectation that we can do things to mice or dogs or primates and learn something about how humans will respond is an outdated concept based on what we know about science today. And it's helpful to take a step back in time and think about how it is that we ended up in this place. Certainly going back hundreds of years, people have been probing animals, try to learn something about the basic anatomy of all kinds of species, maybe trying to extrapolate from one species to another. But the real beginnings of the modern heavy reliance on non-human animals in science is a much more modern phenomenon that in some ways really dates back to some of the atrocities that happened during World War II. So some awful things happened in concentration camps. Despicable things were done to prisoners, and moral outrage rightly followed, but then a wrong turn was taken when the conclusion was drawn that in order to stop these awful things from happening to humans, we should make sure that we first demonstrate safety and efficacy in other species. And while the intention in terms of human well-being might have been sincere, the implications have not been great in all sorts of different ways that we've already been hearing about today. And so if we go back to the early wording of some of these documents that are in the Nuremberg Code and the Declaration of Helsinki, we can see the early origins of the insistence that animal results be determined before we take a look at how compounds might react in humans. And these principles were not necessarily intended to be binding, but essentially they've ended up being quite binding in the way that they have influenced the development of laws around the world. So I'll just mention the United States, where, for example, in 1962, particular legislation emerged that was sort of amplified by what had recently happened with thalidomide. Of course, this is a compound that was given to pregnant women, and their offspring ended up having deformities, missing limbs, and people didn't want things like that ever to happen again. And so the laws were kind of set in place to further entrench the use of animals in drug development. And, if you go back and look at the wording of these kinds of legislative pieces, they don't necessarily require the use of animals, which I think is surprising to a lot of people. And yet the interpretation, the application of these codes has essentially been as if they're required. And I think abolishing the use of animals is going to be as hard as if it were actually legislatively mandated, just because the common interpretation of the laws is such that it might as well be law. And certainly people that I've spoken to in the FDA and whatnot are under the impression that it's not just their interpretation of the laws, but that the laws actually dictate these things to happen. But the larger point is that, these sorts of principles, whether they're entrenched in law or not, these are certainly the codes of practice, and they're heavily entrenched in the way this work is done. And we've got many more options these days. Science has come a long way since World War II and since the 1960s, and we're in the fortunate position of having access to paradigm-shifting methods that make these kinds of laws and applications of these laws in need of a massive refresher. So, with that introduction, let me talk a little bit about the data. So, we've got several facets to consider. One of the primary reasons that regulators emphasize the purported importance of doing animal-based research prior to applying the compounds to humans is to determine the toxicity of the compounds. And so I'll talk about a few memorable examples, which I don't intend as proof of anything, but mostly as sort of anecdotal evidence for which more formal arguments are in the paper in terms of the mathematics of predictive value, and I'll talk a little bit about that too. But I'm just tossing some examples out there by way of creating a general impression here. So one of the more memorable ones is the combination of drugs known as fen-phen, where serious heart valve damage occurred for as many as 30% of people who took this combination of drugs to lose weight. And animal studies had failed to identify any adverse effects to the heart in advance of the rollout to the human population. So some serious life-changing consequences for the patients who took those drugs together. Thalidomide, of course, I've mentioned, widely known to cause birth defects in humans, even though animal models had failed to identify birth defect risks in many species. Vioxx is another one that was all over the front pages of newspapers around the world. So tens of thousands to hundreds of thousands of injuries and deaths among people who took this drug. And, heart failure, serious, long-lasting heart damage to people who didn't die. And this is in spite of the fact that the tests that had been done on non-human animal species had shown the drug was in fact protective of the heart. So completely counter response in humans relative to many of the non-human animals that had been studied in advance. And the spirit behind these examples is captured in a quote from the Handbook of Laboratory Science. It is impossible to give reliable general rules for the validity of extrapolation from one species to another. This has to be assessed individually for each experiment and can often only be verified after first trials in humans. Extrapolation from animal models, like medical art itself, will always remain a matter of hindsight. This is in a Handbook of Laboratory Science telling us that, sometimes it's a matter of hindsight, retroactively cherry picking which non-human animal models worked is a really, really bad way to do science. This is not sound. So this is the situation with respect to lack of safety, not great. And then another primary reason, of course, that animal models are used is purportedly to demonstrate the effectiveness of the compounds. And unfortunately, overall, when you look at the data, we're really falling short on that front as well. So you know, we do these relatively inexpensive preclinical trial tests on animal models with the hope that it is informative about, you know, a good starting point for the human clinical trials. And yet, in spite of that, we're clearly not starting from as sound a position as we would hope, because in each phase, a large percentage of the drugs just don't make it. And so we end up with just a dismal rate of production of new chemical entities. So the rate of production of these new compounds has just been stalled for the last half century. We're just not meeting up to this potential that we're led to believe we're supposed to be in. This golden age of technology is supposed to be leading to all these amazing cures, and they're just not coming through. So when you look at the reasons for these failures in phase 1, phase 2, and phase 3 of the clinical trials, the first reason for the failures, the most common reason, is lack of efficacy, and the second reason is lack of safety. And of course, these are supposed to be the reasons we're doing the preclinical trials on animal models in the first place. So this is problematic. Here's a quote from a former director of the NCI. "The history of cancer research has been a history of curing cancer in the mouse. We've cured mice of cancer for decades, and it simply didn't work in humans." And you know, you don't have to look far. Probably even in this room, somebody's got a computer screen open talking about some promising result, curing Alzheimer's in animals, curing this in animals, promising treatment for, you know, all that ails us in animal models. These are headline-grabbing stories. Humans, we eat up these stories when they show up in the media because it's what we need, but it's just not coming through. In spite of the billions and billions of dollars that are being thrown at this problem, we're just not achieving what has been promised. And it's because the science is lacking. There is no underpinning for continuing down this road of testing in animal models and expecting that model to have good predictive value in humans. So we've seen that lots of treatments have been developed in animals and have failed in humans. So there's been a lot of work done on HIV-like diseases in chimpanzees, for example, and other non-human animal models, and none of them have been effective in humans. We've seen, you know, like 1,000 different drugs that are supposed to be promising for nervous system damage in humans based on non-human animal models for stroke, and they just don't come through in the end. So there's been lots of false hope created, and it's devastating. We should be doing a lot better. And again, I'm talking about lots of anecdotes here, but the formal theoretical arguments are rooted in the mathematics of predictive value, and that makes the general case. So, you know, I'm not just cherry-picking examples that help me make my case. This is just meant to be an overview of the more general case that's supported by the math. We also think about the lost opportunities that arise from us continuing down this failed path that we are so not inclined to abandon. So by opportunity cost, I mean the failure to identify new drugs. So by virtue of investing all of this capital in a failed technology, we're bypassing the opportunity to make other discoveries that might be afforded by allocating those resources to more promising methods. And there are real human consequences from this problem. So lots of drugs that were initially ruled out based on misleading results from non-human animal models would have been lost forever if not for accidental later discovery. For example, based on last-ditch attempts by doctors with near-death patients. There's a story about Dr. Alexander Fleming, who famously said that if animal tests had been required at the time, penicillin may never have been licensed. And in fact, the entire field of antibiotics may never have emerged. Now, this is shocking that, it's kind of by accident, not by reliance on animal models, but happily by accident that we discovered the potential of that compound and the whole family of life-saving antibiotics. So, this problem leads to the state that we're in right now, where we're just not developing safe and effective new compounds at the rate that we should be. So a quote from Nature Review's Drug Discovery: "The number of new drugs that are approved annually is no greater now than it was 50 years ago." And if you take a look at the compounds that are being heavily marketed these days, they're typically older drugs that are maybe being repackaged and remarketed for a new off-label purpose. For example, that's become really common. And the pharmaceutical companies aren't crazy about this. They don't like having to repackage old drugs. They would love to have newer, more effective, safe drugs to be marketing, but honestly, they're being held back. And I was at a pharmaceutical scientists conference a couple of weeks ago presenting work related to these topics, and I was expecting to be the target of some critical commentary, and actually the pharmaceutical scientists in the room, they get this, they understand this, they realize that the animal models are not helpful in the development of new chemical entities, but they're required to do this stuff by the regulators. So they would love for things to change. And, I put the onus on them too. If they want to see these changes, they really need to lobby for them. And, hopefully we'll see that happening more and more. Then there is the financial end of things. So the costs that arise, the monetary costs that arise from our stubbornness and our insistence to continue looking for solutions based in animal models. So again, just some anecdotal information, but sepsis, for which non-human animal models have yielded no effective treatments, are one of the most costly aspects of human healthcare. So in the US hospitals, the cost of treating inpatients is at least $14 billion per year. This is money, surely, that, if we could find effective treatments for sepsis, maybe looking in more promising avenues, we could perhaps put that money to better use. Other costly conditions to treat in hospitals: cancer, heart disease, stroke, lung disease, other afflictions that have been studied extensively with non-human animal models are also costly to treat. And the treatments that we do have still pose lots of risks to the humans, the patients that are undergoing these treatments. And then, these treatments that are in existence, to the extent that they're effective, they're the best that we've got access to, and so we use them. The cost of administering those treatments reflects the cost of continuing to try to develop new chemical entities, many of which are failing. So the pharmaceutical industry still has to pay for all those human clinical trials. It's actually the human clinical trials that are much more expensive for them to conduct than the animal-based testing. But the clinical trials can't be done until the animal tests have been done, and they have to build on the animal results, which of course are not predictive for human responses. So much money is being dedicated to this enterprise with high failure rates and lack of great success. And we need to think about whether it makes sense to continue directing those dollars to a failed enterprise. Obviously, I think it doesn't make sense. I think that there's a better path forward. So let me talk a little bit about the math. And thinking about it now, I probably shouldn't have said that I was going to talk about math, because nobody loves to talk about math. But it's not really math. I'm just going to wave my hands around. I'm not going to put any formulas up on the screen here. I'm just going to talk generally about the concept of predictive value, which I've been making reference to in kind of casual terms. And all I'm talking about here is what medical professionals called the gold standard. and the idea of true positives versus false positives when we talk about a test, and true negatives versus false negatives when we talk about a test. And so the gold standard is, in this context, what will the outcome be in humans? Once we've got a compound that we're thinking about marketing as a drug, let's say, we want to know what will the response be in humans. And we try to figure that out by doing tests in animals, and we hope that if we find something is safe and effective in animals, that it's going to translate to be safe and effective in humans. And unfortunately, sometimes we think it will be safe and effective in humans based on the animals, and it turns out that we're wrong, or we think it won't be, and it turns out it would have been safe and effective in humans. Sometimes we then abandon those things that didn't work in the animal models, and we find out accidentally many years later, or maybe never at all, that it would have been a promising avenue to apply in humans, safe and effective. And we don't know how many compounds we've lost to that dustbin of history. And so when you look at the actual numbers, what we find is, in terms of predictive value, animal models are not overall predictive about human responses to drugs and disease. When I say not predictive, I mean we're better off flipping a coin. It's pretty bad. That is really bad. We shouldn't be flipping a coin to figure out what's going to work in humans. So the predictive value for responses to drugs in development are less than 50% overall. And, you know, people will sometimes cherry pick, like, well, but what about this one? This one worked. And, you know, of course, in retrospect, we can always point to things that worked. But if I told you, I'm going to predict the weather tomorrow in Baltimore, I'm going to flip a coin, heads it's going to be sunny and tails it's going to be cloudy, and I happen to be right, you wouldn't say, that is a magic coin, let's keep using that coin, right? Like, of course, every incorrect method can occasionally be right. You know, just because something is once in a while correct, it doesn't mean it's good science to keep using it in general. So we want to think about things in terms of predictive value and make sure that we're doing good science. So this is the point. We would be better off tossing coins to pick which drugs should be marketed than basing the decisions on results from non-human animals. And it's pretty sad that this is the state of affairs. So let me talk a little bit about the theoretical foundations. So putting aside the anecdotes and the data and the math, just some scientific reasons why non-human animals just simply cannot be expected to predict human outcomes. And to talk about this, we just need to refer to a little bit of complex system science. And so we can think about a simple system, something where we can easily replicate what the output is of a little experiment. For example, like, let's say we're looking at rolling a perfect sphere down a ramp with no friction in a vacuum. We can keep doing that over and over. We're going to know exactly how long it takes for the sphere to roll down the ramp. There's nothing complicated about it. There's nothing complex about it. It's just simple. We know exactly what the inputs and outputs are. There's no randomness. We can predict each time exactly what's going to happen. And biological systems aren't like that. We're complex. We're comprised of many parts that interact in ways that are unpredictable. We've got hierarchical levels of organization. There are feedback loops in complex systems. They self-organize. They react dynamically and unpredictably and sometimes randomly with respect to the environment. There are nonlinearities in their responses. We could say the whole is greater than the sum of the parts. We can't just put together a bunch of blocks and generate a complex system. And importantly, they're dependent on initial conditions. And I mention this because different species you could think of as having different starting conditions when we look at how they're going to respond to drug stimuli, for example. And this is a little excerpt from the chapter. Animal modeling seeks to use one complex system, be it a mouse or a monkey, to predict responses to perturbations that occur at higher levels of organization of another complex system, a human. To do so ignores the most basic fundamental features of complex systems discussed above. Given those features, it is outside the realm of science to use one complex system in expectation of it having predictive value for another when the perturbation affects higher levels of organization. So there might be instances when you can look at, a very simple level of comparison, like a cell in one species and a cell in another, or a neuron in one and a neuron in another, and at that very simple level, talk about commonalities. But when you start thinking about the entire biological being and exposing that being to a stimulus like a drug or a disease, and you try to predict how a different species will respond to that same stimulus, you're in Never Never Land. You just can't expect two different complex systems to respond in a similar predictive way. It's just beyond science to have that expectation. It's not supported by the science. So, trans-species extrapolation is possible in these simple scenarios, like comparing a neuron to a neuron, but as soon as you get up to the gross level, when these perturbations concern higher levels of organization, one evolved complex species will not be of predictive value for another. And thus, using dog models or mouse models to predict human responses is not going to work. Predicting dog responses based on other dog responses is getting closer. But honestly, even for two individuals within the same species, small differences in DNA can lead to completely different outcomes that can mean the difference between life and death. So, for example, tiny little differences in one amino acid in the human chromosome determines whether or not someone suffers from sickle cell anemia. And so having a large overlapping percentage of DNA can be largely immaterial in trying to predict responses to perturbations when we're talking about complex systems. Continuing on this theme, complex system science aside, the second point is evolutionary biology. So small changes in genes over long periods of time result in new species. So mice are the outcome of an evolution that split from humans many, many tens of millions of years ago, and they've evolved immensely over that period. So, humans have also evolved immensely over that period. So from these different origins, we've evolved to be quite different. And even though different species may share common genes, there can still be vast differences in the way each species responds to different stimuli and perturbations. So, the progression from HIV to AIDS, for example, which is commonplace in humans, is not typically seen in chimpanzees, even though we have a lot of overlap in our DNA. So just generally speaking, different species may or may not have common reactions to drugs and disease. We're not able to predict when the reactions will be common across species due to complex system science and evolutionary biology. So we want to think, what can we do instead? What are the better options? And I just want to make reference to the streetlight effect, an anecdote that's sometimes called the drunk's search. When I was presenting in Washington, D.C. a couple of weeks ago, I wasn't the only one using this graphic. It turns out there was another presenter, a scientist, making the same argument with the same graphic. So if you're not familiar with this anecdote, a man has been at a bar all evening and he's intoxicated, and he comes out of the bar and he can't find his keys, so he's poking around underneath the streetlight looking for his keys. And a policeman comes along and says, "Sir, what are you doing?" And the man says, "I can't find my keys." So the policeman starts helping him look. And after a few minutes, they're not finding the keys, and the policeman's getting frustrated, and he says, "Sir, are you sure this is where you lost your keys?" And the drunk says, "Well, no, actually, I lost them somewhere else, but this is the only place where there's light," right? So this is the state of pharmaceutical research right now. We're looking under the light, but the answers are not under the light. This is just where the light is. It's the easy path to just be using the animal models. The answers are somewhere else, out there in the dark, and it's not a good excuse that there's light right here for us to keep looking here. We need to be creative and innovative and use all our human potential, put that to work, finding a way to look for things even in the dark. And that's my hope, that we can make use of some of the modern technology that's come along that will allow us to not be like the drunk looking under the streetlight. So some of those better options are very promising. The idea of personalized medicine, seeking druggable targets using human tissue instead of using non-human animal tissue, linking genes to diseases and designing drugs around the proteins produced by those genes, microdosing to determine safety and efficacy instead of testing on animals. This was mentioned in the last talk. So this is the idea of applying nanogram quantities of a drug measured in billionths of a gram, which is safe, and administering that to patients to evaluate the safety of the compound. This is science. More on personalized medicine, the idea of matching a patient's genes to her disease and drug response. You can account for the fundamentals of molecular biology, different molecular biological differences in the genetic makeup across individuals. We've come a long, long way since the 19th century, when animal models thought all species were just scalable versions of each other with the same physiology. Now we know about the cross-species differences that exist, and also the significant differences between the sexes, across different races, and even between identical twins. We can have markedly different responses to compounds in two people who were born genetically identical. Epigenetics maybe plays into that. It's just, there are lots of scientific reasons why we can't expect one individual to have the same response to another, even within a species, and then we're just completely in deep water when it comes to the cross-species extrapolations. We've heard of organs on a chip and bodies on a chip or humans on a chip. This was a buzzword at the Pharmaceutical Scientists Conference I was at last week. These are all very promising. They're not panaceas. There are challenges in some of these methods. Organs on a chip and mathematical modeling are built on strong assumptions. And to the extent the assumptions that go into building the model are not sound, we can end up with outcomes that are not sound. And so we just want to make sure that we're applying rigor to these methods as we move forward. But again, we can't keep using broken methods, by which I mean the animal modeling, to look for answers when we know that those models do not have predictive value for human outcomes. So that's the first chapter. My discussion of the next chapter is briefer because it is simpler in many ways. And so this is chapter 2 of the book, How to Evaluate the Science of Non-Human Animal Use in Biomedical Research and Testing, a proposed format for debate. And in this chapter, we are talking about how to move forward, how to resolve this challenge before us. Animal advocates and some scientists don't necessarily agree about some of the principles that we're discussing at this conference today. And what are some of the obstacles that stand in the way of us reaching consensus? Now, I'm a behavioral economist. I'm an economist by training who does work on how human psychology influences decision-making. And so some of the things I think about are, what is it about our human nature that makes it hard for us to shift course? And there are lots of things that get in the way, a few of which are particularly problematic in this context where we're talking about scientific arguments, we're talking about science. Many lay people are intimidated by science, and they're inclined to give way to the experts. So people who have scientific training can be very persuasive when talking to members of the public. And some of those scientists might have entrenched conflicts of interest and be wanting to promote the way things are are being done now, and members of the public might be inclined to just defer to them. So obedience to authority can get in the way of progress. Status quo bias is a deeply entrenched feature of all of us. It's a fancy way of saying humans are lazy. We like to do what we've always done, and ideally we like to do nothing at all. So if we're already sitting, we'll just stay sitting, thank you very much. And if we're using animal models to do, scientific research, let's just keep using animal models. Seems like it works, right? So status quo bias can be hard to overcome, often unless we have something more promising that gives us, good results that overcome that natural inclination to be lazy. It can be a really tough one to overcome. Confirmation bias is a big one. We always like to think that we're already really smart, and we go out seeking information that reminds us how smart we are and that we're doing the right thing already. So, it can be very tempting to, when you're trying to promote the continuation of a course of action, to go out and look only for feedback that reinforces your preconceived notions, and to discount any evidence you might come across that contradicts what you want to believe. So this is an obstacle. And then conformity, or the inclination we all have to give in to social pressure. So admit it, when you heard in the news last year that Bitcoin was going up and up and up in value, and you were hearing about people who were making money, you were tempted to maybe jump into that market. And it's kind of a fear of missing out that people have, and wanting to cave in to be accepted by peers and emulate what they're doing and replicate what they're doing. And this can play a role in contexts like this as well. If something is the norm, it's really difficult to develop practices that deviate from the norm. So, in the context of trying to change this paradigm, we've got a lot to overcome. And, it doesn't help that researchers are sometimes not just prone to these kinds of effects that I'm talking about, but sometimes they're actually not even being truthful about the validity of non-human animal modeling. And maybe it's due to having entrenched conflicts of interest, or having been persuaded by this confirmation bias. It's hard to disentangle why these things happen sometimes. But here's a quote from Animal Models in Toxicology, the preface of the book. Biomedical science's use of animals as models is to help understand and predict responses in humans, in toxicology, and pharmacology. By and large, animals have worked exceptionally well as predictive models for humans. I just don't think that's true. That's just categorically not true. When you actually look at predictive value, it's very poor. It's not accurate to say that the animal model has worked exceptionally well. You can point to instances where there have been successes, but it's been on an ad hoc basis here and there. And when you look at the results overall, the overall predictive value has been quite disappointing. From the Handbook of Laboratory Animal Science, an important group of animal models is employed as predictive models. These models are used with the aim of discovering and quantifying the impact of a treatment, whether this is to cure disease or to assess toxicology of a chemical compound. So again, I would argue that while they're being employed as predictive models, their predictive value is poor, certainly insufficient to justify the continued use. So in this chapter, we therefore aim to put forward a format for debating the issues. And we model our proposal on the well-established practice of peer review in academic journals. And we promote the idea that these are the kinds of questions that should be considered in the debate. So are claims being made by advocates on either side consistent with modern scientific understanding of the issues? Are the assumptions being made valid? Are the methodologies being employed appropriate? Are the statistics that are being used, were they calculated correctly? Is even the rational basis of the logical argument sound? Are people bending the rules of logic in the premises that they're putting forward? And are the findings of interest to the scientific community, policymakers, and or the general public? given every single one of us has a stake in the pharmaceutical, in the development of drugs and the treatment of diseases, I think we can agree that these issues are of broad importance, and so we would like to promote the idea of the issues being debated and settled. So the specific format that we propose is to examine the subject whether non-human animal models have sufficient predictive value for human response to drug and disease. Ray and I, having written the first chapter I was talking about, we feel strongly that the answer to that question is that no, they do not have predictive value. But let's have it out. Let's hash out the issues in a forum that's public and transparent. We suggest that it be sponsored by a government or major science organization. In the UK, there is a movement to have a debate just like this. The Parliament has been talking about these issues in the last few months, and there's a movement afoot to have a debate like this. So it could be a government like the UK that would sponsor it, or a nonprofit organization, or a scientific association. There are lots of possibilities, but the design of the debate, we would ask meet certain specifications to ensure impartiality of the parties that are adjudicating. As an incentive, there could be implications for future funding and legislation. That wouldn't be up to us to decide. Of course, people would need to come forward to facilitate that. And then we would have the debate being moderated, and each side would be represented by a recognized expert who may consult with other experts for guidance and input. This wouldn't be something that would just happen, in an hour. There would be a period of time over which some preparations would need to take place. And we suggest that there be a panel of somewhere between 1 and 2 dozen expert scientists representing some key areas in science to assess arguments. And we put forward some suggested areas in the chapter representing, biological sciences, physics, various areas, statistical analysis, to assess the merits of particular arguments as they come up in the debate. And we also put forward some proposals for how to assess reduce the number of conflicts of interest that would exist in these individuals participating. And so basically that's the proposal for the debate. I refer you to the chapter for more details, and it's a modest proposal that we think is achievable, and it could, in principle, be a face-to-face debate, but given the complexities of the issues and the difficulty people might have in assessing the arguments quickly, we actually propose that the format for the debate be in writing, and then it could take place over a period of time, including perhaps more interactive input from experts from different areas. So that's our modest proposal for a debate. And if I could, I think I still have a few minutes, I would just mention a piece that is related to these 2 chapters, which were really the impetus for some of the ideas that led to the creation of the chapters. A piece that Ray and I just published in Business and Society Review a few months ago, where we're looking at the validity of using non-human animal models from a human ethics point of view, and particularly, everybody's a stakeholder of the drug discovery process, certainly patients, also clinical trial participants who are right now volunteering their time and their bodies on the basis of often flawed results from animal models, shareholders of pharmaceutical companies, employees of pharmaceutical companies, taxpayers, donors to nonprofits and universities, future generations of scientists. These are all stakeholders in the drug development initiative, and we argue that their interests are of great relevance and that they're being harmed from a business ethics point of view. So basically, constrained by these standards that I mentioned that date back to World War II, the industry is being required to employ animal models, and this is preventing the development of harmful drugs, and it's hindering the development of useful drugs. And if we could modify the way discovery is occurring in this enterprise, we feel there would be great benefits to all these stakeholders that I just listed. So the patients, the clinical trial participants, the taxpayers, the shareholders, the employees, the researchers themselves. So that's our goal. And that paper it's free access. Actually, the publisher is not charging for download, and it's available. There's a link on my website if you want to look it up, or you can email me, and I will be glad to send you the link as well. I've cited a lot of different pieces. I think I didn't cite anything that isn't cited in one of the chapters, so you can dig up any of the citations if you're interested. And I'm really, really grateful for your time and attention. It's been a pleasure to be here and to have contributed the chapters, so thank you.
Thank you, Lisa. That was wonderful. Any questions from the audience?
Hi, thank you, Lisa. I have enjoyed all the great media coverage of your new article and your recent presentation, so congratulations.
Thank you.
It seems to me that a real driving force, given the political capital they have, could be the pharmaceutical industry, because they're absorbing most of these costs of the waste happening even on the basic research side that they're not involved in. And I don't know, in your experience as a finance expert and experience at that conference recently of pharmaceutical sciences, if you think there is that interest in the industry in being a leader in pushing this issue, not only for the required testing, but all the testing that happens, the basic research that happens before that NIH is funding and flushing money down the drain and misleading the pharma companies down paths that they're wasting billions of dollars on.
Thanks for your question, Justin. So my sense from the representatives I've interacted with from the pharmaceutical industry is that this isn't news to them. In a sense, they're quite aware of the poor predictive value of animal models. And my impression is that they just feel bound right now to adhere to regulations and produce the data that is required by the FDA, for example. And I've heard anecdotes of startups, for example, that have developed small tweaks of kind of known safe and effective compounds, but the tweak was sufficiently far enough away from the original compound that the FDA has told them, even though the compound is like basically ready to apply straight to humans, the FDA has told them they have to actually go and backfill with more non-human animal data before they can even apply this, safe and effective compound to human trials. So there's a lot of frustration I'm sensing, and awareness certainly. And I guess the next step will be to help them understand that, in a sense, they've got a lot of control and at least power to lobby for change, and I mean lobby in the formal sense. So, I would love to see them mobilize and use their collective weight to push these issues forward. And I suppose armed with, contributions from scientists, the sorts of chapters that we've seen in this book, that can give them the kinds of resources that they need to start to put forward some formal arguments, and then combined with their monetary incentives, they have the incentive to develop drugs that are going to be successful in clinical trials, because then they can market them. They don't like having to run clinical trials that are ultimately losing money for them and failing to lead to the development of useful products. So I think there's an untapped potential there right now. I don't know exactly how to harness it, but I'm quite compelled to, put my head together with other people to think about how that might be achieved.
I mean, they're one of the most powerful lobbies in the world.
They are, yes.
If they want this, they'll get it.
Exactly.
It's a matter of them prioritizing.
So let's help them.
Thank you.
Thanks.
Hi, Lisa. I'm not a scientist, but my job is to translate scientific research for a lay audience. As a writer, so I'm particularly interested in language. I saw here that says that one of your slides said that non-human animal models cannot predict human outcomes, and the "cannot's" even italicized. Mm-hmm. Do you think that's a little too strong of a statement? Is that an accurate statement? just as far as someone like myself that would attempt to take that and then talk to a lay audience about the research? Thank you.
Thanks for the question. Yeah, it was italicized on purpose, and I mean it in a statistical sense, predictive in a statistical sense. So, and I would bring up that little sort of silly example I gave about the coin flip again. You know, like you can predict the weather tomorrow using a coin flip, but it's not really predicting. It's an accidental random correlation. And so I would argue that we've had accidental random correlation where we've happened to have seen a response in a non-human animal model that correlates with what we end up seeing in humans, but we had no theoretical or scientific basis to expect that correlation. It's not predictive in a scientific sense. And that's what I mean when Ray and I say that animal models cannot be predictive. In a statistical sense, when you look at the entire paradigm of using one species to predict response in another, there cannot be predictive value.
Any more questions?
Is there any reason to believe the alternative? I'm sorry. I enjoyed your talk, but I'll be the devil's advocate. Is there any reason to believe that alternatives to animals would perform any better than a coin?
I think personalized medicine is already offering better outcomes in terms of predictive value than non-human animal models. So, you know, there are applications in treating some kinds of leukemia, in treating some other diseases where we're using, you know, the human's own genetic makeup to make a treatment that will work for that human. And it wouldn't work for their sibling necessarily, but it's tailor-made for that individual. And that is a much more sound path forward in terms of predictive value. So I do think that the promise is already there in some of these newer methods, yes.
Thanks a lot.
Well, in 2009, something of great value to us here in the United States happened, and that's when Professor Hartung came to Baltimore and CAT. And since his presence here, he's continued the authoritative movement toward alternatives for animal use in the name of, or for the benefit of human welfare. Even a brief look at his awards and publications, which are repeatable, I'm certain. He's an extraordinary scholar and scientist. So, Thomas.
So this was by far the most flattering introduction I ever got for a presentation. And I have to live up to this, which is not that easy. Let me see. And much easier with my slides than without them. Good. Okay. Yeah. Good. Here we are. And now. Okay. So, if you look backward to this day, I think this was altogether pretty depressing. Yeah. I mean, we have seen in all of these talks, one after the other, animal experimentation sucks. Yeah? And I have to test, by large, all the facts and all these things which were put together are correct. It's my experience after 30 years in this business is that's the situation. We have also seen that the adaptation of new is rather slow. Despite this, the majority of scientists believes in animal models. I see regularly our grant applications refused because we have no animal part to control our findings. We don't see really a dramatic change, and most of my colleagues believe you need a knockout mouse to produce a new line for a textbook in medicine. So this is a situation where we need a revolution. And we know from Thomas Kuhn about the nature of scientific revolutions. that essentially you need two things. You need doubt in the current paradigm, and you need the availability of new technologies. I think you have shown very nicely that there is the doubt increasing in this paradigm. We have the hard facts. I will come back with some more of them. Some are a bit overlapping, and I'll do this quickly. But I would like to bring us in this last presentation, let's say in front of last presentation actually, I would like to bring in a more positive spin. I would like to talk about the technical opportunities which are actually coming to show you that we might actually be able to do better. We're up against an enormous number of animals used worldwide. You know from my accent and from the introduction that I'm European. I'm quite fascinated about the differences in approach in the US and in Europe. Europe has about 550 million people, US 300. The number of scientists in percentage is about the same. So we have almost 1.5 times more scientists there, 1.5 times more scientific publications in Europe. But in the US, we use 6 times more monkeys, and we use 4 times more dogs to produce a similar output. Obviously, a willingness much easier to go into these animal models. We also see that at least until a few years ago, there was essentially no funding for alternative methods here in the US. When I was working as head of the European Center for Validation of Alternative Methods, I sometimes felt like an alien when I was coming here, talking about a world where alternative methods receive hundreds of millions of funding to be produced. This has changed, fortunately. The agencies here in the US have taken a very active role. We saw a bit of their roadmaps, but about 300 million has been spent in the last 5 years on organs-on-chip models in the US, about 200 million on robotized testing, things which were not foreseeable when I moved exactly 10 years here to the States. And this is because it comes here in the US not as an ethical debate. It does not come as an animal welfare debate, but as an economical and technological discussion. And there's good reason for this. Because we know far too little about our chemicals. Most people believe by buying a product, you actually get something which has been sufficiently tested to be on the safe side. However, chemists have produced millions of structures. About 140,000 are in products worldwide. About 1,000 come to the US market every year. At least this is the number of pre-marketing notifications. I just learned only about 50% of them actually make it to the market. Let's say 500 every year. So, and what do we know until now? Only 1 in 8 of these substances has any toxicological data in their pre-marketing notification. So the majority comes on the market in the products without any testing. Take the area of food additives. In the US, we have 4,500 food additives. For more than 80% of them, there's no public data at least available. So things which we are really consuming everywhere. We have e-cigarettes. Over the last 10 years, they were introduced. 7,800 flavors were registered in 2014. None of them really tested for this use. So we are really living in a world where we cannot address this with traditional type of testing. 3% of all of the chemicals in the market, roughly, were extensively tested. 10% have some type of data, but very often these are just acute and topical toxicities. And even after finishing the enormous effort in Europe on the REACH legislation, we are moving now to about 8% extensively tested chemicals if the testing proposals are being executed. And about 16% of the substances have some type of test. So there's a tremendous demand to do something. And toxicology is an economical challenge. Our earlier studies from 10 years ago estimated 3 billion of animal testing costs spent every year. One pesticide does cost 20 million of toxicological testing. You have seen about 30 animal tests which are done which form this package. A cancer assay alone is $1 million for one substance and one species. And this is probably what every consumer would like to know. Is the stuff I'm eating producing cancer? We have only about 3,000 substances tested for cancer. And the assay is not really a very, very helpful tool because 53% of these 3,000 substances tested are positive, even completely harmless stuff among it. 23 ingredients of coffee tested positive in the cancer bioassay. And it takes about 5 years to get these results. And you need about 20 kilograms of substance to feed animals for 2 years. Tell a chemist, I would like to have 20 kilograms of the substance which is not yet going into product, just to test it in rats, in order to get, after 2 years, with 50% likelihood, a positive result for a cancer bioassay. It is ridiculous, yeah? So this is simply not adequate for the purpose. And I really believe the most important omics for us is economics. We have just published these data here, an update of the similar paper 10 years ago. And what I have, however, found most remarkable, in the meantime, in vitro toxicology is itself a market of 14.4 billion, and in vivo has increased only from 3 to 4.2 billion in the same time. So there is technology coming in. And there's a lot of very interesting data in this. We took them from about 150 websites and many, many articles in order to just illustrate how the various sectors are doing and to show that animal testing is itself an industry which is also often the roadblock for change, because whenever you come with a disruptive technology, there is a business being disrupted. Animal testing CROs are in the $50 billion range as an industry themselves, and they are very often up against us. And if you just see the shopping list of animal tests, yeah, many of these animal tests are in the several $100,000 range. So you could, instead of running one chemical in one of these assays, you could sponsor a PhD thesis or two. A lot of more information you would get about the respective substances. So it is really about, this is no longer the gold standard. And by starting to doubt, we are actually beginning the scientific revolutions. And we need to illustrate this, we need to communicate this. And one example, it was actually referred to 10 years ago, I put data together about aspirin, just to show that aspirin would fail essentially every possible test which we were applying to new medicines. And this is important because most people are only asking, are we sufficiently protected? But this is also about, are we sorting out valuable medicines because of assays which wonder drugs like aspirin cannot really pass. So it's a very important aspect to keep in mind. If we are not using human relevant data, we are also killing people by simply not giving the right information and not getting this to the market. This is not to say we should stop regulating. The tossing of a coin analogy I would not accept. I believe that we are increasing the odds in the development process of producing a drug. And drugs have helped us to triple life expectancy since 1850. Drugs have helped us, and other safety tests have helped us to make this place a safer world, but at which cost? But we should also be careful, we have to regulate industries. And this is an advertisement of Bayer on aspirin, as you can see here on the top, in the beginning of the last century. But there's a second very interesting product by Bayer, which is heroin. Only 2 weeks after Felix Hoffmann synthesized aspirin by acetylating salicylic acid, he acetylated morphine and produced heroin. And it was marketed as a sedative for coughs. It became very successful, but not in the pharmacies. So we should give it a look what companies want to bring to the market. It is really about finding the right balance between the need for safety, the need for proper drug development, and what are the means which are bringing us there in an efficient way. This cartoon here says, we thought you'd like to meet Reggie. He's the rat who we experimented on to find a cure for you. If you would confront patients with these animals and say, that's why we are now believing we can treat you, they would certainly say, really? It is really a shaky foundation on which our wonderful clinical research takes place. Animals are not very good crystal balls for the safety and efficacy of drugs. 97%, according to the statistics drawn from the FDA approvals and registered trials, fail. And these numbers are probably still inflated because they include tests on substances for new purposes where we already know they're at least safe. They include substances which are Me Too's, generics, which are just the same substance or something very, very similar where we already know that they have the same active principle. And even afterwards, we still have to withdraw about 8% of the drugs because of side effects which we did not anticipate even throughout the entire clinical development process. So there is something wrong in the state of Denmark, or similar as Hamlet has said. Okay, but there is increasing awareness. What you voiced today, you can hear nowadays in Nature and in Science. And this was unthinkable some 10 years ago. The reproducibility crisis, which is acknowledged, which is mainly an animal experiment reproducibility crisis, is something which made it to at least strong awareness. In this article here, earlier this year, in Altex, I think I have the full citation a bit later, we did show for the systemic toxicity of cancer, reproductive toxicity, repeat toxicity, a lot of the shortcomings and our vision for moving forward. But the important fact is for all of these, there's only about 60% correlation between mice and rats. There's no reason to assume that any of them is predicting humans better than they predict each other. And this, I think, is a very powerful message. Yeah? And we are not 70-kilogram rats. I wrote this 10 years ago and has been frequently cited since. It's a very simplistic view, but that's what it is about. We are not 70-kilogram rats. And you can see here, the weight is not fitting, the age is not fitting, and the patients for which we are developing drugs now have survived one cancer, are obese and diabetic. That's nothing you can see in our rats. Yeah? And they are on 6 meds or more, so there's a lot of things to consider which we cannot model in these systems. But we wanted to get hard facts. I have a chair for evidence-based toxicology. We want the facts to speak. In toxicology, we should actually have the best animal tests, because these are the only tests which are carried out under a quality assurance scheme, good laboratory practice. They are only done by skilled performers who have shown before that they are capable of doing these tests, not the youngest students like in our labs. And we're using strong doses, strong effects, maximum tolerated. And we don't need a disease model to test for testing pharmaceuticals. It's just healthy animals. Boom, hit them. We want to know how good this is. So what we did 2 years ago is we produced the, at the time, largest toxicological database by downloading the registration data from the European chemical legislation, ECHA. And we found at the time 10,000 chemicals with 800,000 associated toxicological studies. 800,000 toxicological studies. And we not only downloaded it, but the important part was by natural language processing, we made them machine readable. Because these are largely PDFs or text which is not standardized. So Tom Lichtefeld, who in his PhD did this work, he programmed about 500 pages in order to teach the computer to read that eye irritation corrosive is the same like category 1 or cat.1, and an Arabic one is the same like a Roman one, and other ways of expressing this. But this work then led to a machine-readable database, and in this database we had incredible repetition of experiments. We found 2 chemicals which were tested more than 90 times in rabbit eyes. We found 69 chemicals which were tested more than 45 times in rabbit eyes. On average, every chemical registered was tested 3 times in rabbit eyes. There's good reason for this, because data before was simply siloed, not public. A company could not know that there is possibly other data available. And before the mutual acceptance of data, you had to do the experiment in the country where you were registering. So I don't blame companies for producing this redundant data, but I thank them for making them now finally available because of the provisions of the legislation. And what is the outcome? We found for 6 tests, the classical 6 packs of toxicology, which uses 57% of the animals in toxicology in Europe. So really big bites, about 600,000 animals per year. We found for them 350 to 750 chemicals which were tested more than twice, as you saw, up to 100 times. Yeah? And we found them, on average, 81% reproducible, which is, however, inflated by the fact that a non-toxic substance will always be non-toxic. It does not turn suddenly toxic. But the toxic substances were only 69% reproducible because they sometimes don't show. And this shows the tremendous safety gap which is left by these type of methodologies. But this is giving us a benchmark based on really not a few substances, but on a tremendous area of chemicals, representing a large part of chemistry. It shows us how good do we have to be in order to offer a solution which should be acceptable. So in this approach, moving us into big data also allowed us to move further and develop predictive tools. And these predictive tools are based on this preliminary work, the study which we reported in 2016, and it's natural language processing. This is Tom. We created applications, web applications. He also formed the company ToxTrack in parallel in order to make this feasible. And we teamed up with Underwriters Laboratories, UL. That's an organization which produces safety standards and also tests against it. It was created by an act of Congress at the Chicago World Exhibition. And you find their seals on every toaster and electrical appliances and whatever. And for full disclosure, I'm consulting them, and also I'm benefiting from sales of products which hopefully will soon make it and generate some profit. Okay. In the meantime, we created with them a database of 10 million chemicals. 300,000 of them have biological data. In the meantime, 50,000 have animal data. An enormously rich database. And this database allowed us to produce really a revolutionary approach from my point of view, using big data and artificial intelligence, machine learning. We described our visions in our Food for Thought articles and others, so you can read all about these developments, and I don't want to waste time on this. But I love at least this quote, which is by Dan Ariely about artificial intelligence and big data. He says, big data is like teenage sex. Everyone talks about it. Nobody really knows how to do it. Everyone thinks everyone else is doing it, so everyone claims they are doing it. So here's our claim that we are doing artificial intelligence and big data. So what we did, I will be brief here. We combined actually 2 approaches. One is called read across, an approach where you only look for similar chemicals to make a prediction, say, I fill a data gap by saying every chemical around this has this property or does not have this property. So why should my substance have a property suddenly? It's a very pragmatic approach, very much used in the REACH legislation, but it is done by an expert who happens to know a few chemicals, has access to some data, and interprets these. And there's computational approaches which are trying with a computer to describe the molecule and make a prediction for parts of the chemical universe. So we call this a RASAR, a read-across-based structure-activity relationship. So, and what we needed for this is to create such type of map, a map where similar chemicals are close to each other and those which are far from each other don't share chemical structures. We did this for the 10 million structures, and it did take an Amazon cloud server 2 days to crunch these numbers. Yeah? So this is several $1,000s of computing costs to produce such a map. But now we can identify for each and every chemical what is the neighborhood, what are the properties of this neighborhood. And we entered 74 different properties into this. And you can also, for structures which are not among the 10 million, you can place them with some calculations inside the map. It is still about 500 million calculations, but that's nothing nowadays. And you can make predictions from this. So this is how the computational map actually looks like. You really have to zoom in. This is 10 million structures. Yeah? Now you start seeing them. The distances are shown here by the lines. And then you can start entering data. You have positive, negative, for each of the different tests, chemical, physical properties. So this was an enormous effort. And then we started to develop a technology which we call data fusion. It's transfer learning for people in the field of artificial intelligence. So we said, why should we make our predictions only by, let's say I have chemical here, and I want to know whether there's an irritant. I look into the neighborhood, but why should I limit myself to looking for the information on eye irritation? I might have no information on eye irritation, but I know it's a skin irritant. And if it's a skin irritant, it's likely an eye irritant. If it's a mutagen, it's likely reactive chemistry. It's informing us that it will be likely an eye irritant. And then we let the computer do its work. So, in the meantime, we have done this for 9 tests, animal tests. So we added acute inhalation and acute and chronic fish toxicity. You see here the number of chemicals for which we have classifications. This is really in the 10,000s up to almost 50,000 chemicals for which we have eye irritation data. An enormous database, yeah? And then we do predictions, and I don't want to request too much of you from this data. I can simply say, for 100% of these chemicals, 190,000 examples where there's a chemical with a toxic classification or non-toxicity classification, we were 87% correct. So compare this to the 81% reproducibility of the individual animal test. This is the moment when Deep Blue did beat Sakharov in computer chess. It was the first time that the biological system was not as good as the computational one. We published this in July. It was covered the same day in Nature and in Science, and in the meantime, in more than 110 other articles in The Economist and the Financial Times, and name them, the Chemical Watch and Chemical World, and all of them. So in summary, I think this is a very nice example that we are moving, for example, with technologies from the computational side into areas, and this is only the beginning, where we can be better than the animal test. And we have ourselves already optimized from this stage, and we're going to make the new publications now about the optimized models, and other people with whom we have shared approaches and databases are working on this to make it even better. But it is, I think, for the first time, a pretty clear proof that you can be as good as the animal, or as bad as the animal, as we have to say, because unfortunately we're not interested really in animal toxicity. But we're working at the moment also on the prediction of human data. Emily, sitting here, our PhD student, is just completing something on this in the area of skin sensitization, where we have human data for 450 chemicals. But we have to take this all with a grain of salt. This is not yet validated. This was controlled only in our lab. If it is really that simple in the future to press a button and possibly substitute 600,000 animals in Europe, more than 2 million worldwide for all of these toxicity tests, it would be too good to be true. And this is unfortunately often the case. I really would like to see this. I've seen as a head of validation bodies many things collapse, which will look very promising. But still, nothing has had preliminary data with 190,000 chemicals so far, delivering this type of results. But we have clearly a situation, trash in, trash out, every function of trash is trash. We're working with animal data. We don't work with the human data here. And we have very questionable quality, as I only showed you a little bit earlier. Yeah? We also have a lack of public animal data for the really interesting endpoints like chronic toxicities, cancer, reproductive toxicity. There's a lot of problems around this, so we will still need testing. But it could be a good bite of the overall cake of animal testing. So we are not 70-kilogram rats, but we are also not 70-kilogram cell culture. But we are coming closer with the modern type of toxicology or modern type of cell systems, which is the organotypic cultures, the organ-on-chip type of things. And I will give you our example of mini brains to illustrate what is happening at the moment as a revolution in the way we do cell culture in the lab. Our interest in the brain came from autism. You might know how autism rates have been dramatically exploding in the US. This is 1970, these are CDC data. And you see the increase until 2013. And in April, we got the 2014 data. It is now from 1 in 68 children. In only 1 year, it went up to 1 in 59 children. Dramatic increase. And I very much believe that society at some point will want to know from us, what do chemicals do to contribute to this? It's very similar to the endocrine disruptor discussion, where fertility decreasing, the people want to know, are chemicals doing something? So this will be somehow the endocrine disruptor program of the next decade. There's certainly other things, but genetic can't be, at least not alone. And we have done a lot of work on this. This was one of our, let's say, articles around the topic. We have been holding so far 4 international conferences and 8 workshops on developmental neurotoxicity, together with my previous shop, Equam and CAT. And we will be hosting in Germany in early 2020 the 5th International Conference on Developmental Neurotox. Again, an example where the animal experiments are useless. If you want to test for developmental neurotoxicity, there is an EPA guideline, there's an OECD guideline. It does cost you only 1.4 million per chemical. It consumes 1,400 animals. And about 200 substances have been tested, but not a single one led to a regulatory decision. So not even the EPA is requesting this anymore. But we need a tool. And for this, we developed what we call a mini brain, or brain spheres now, to be more precise. These are balls of cells. They are about 350 micrometer. We can produce several hundred or lower thousands in a week, in one batch. So one 6-well plate in the lab is about up to 800 mini brains. They are very, very standardized in composition, size. The neurons are spontaneously communicating. To show you this, perhaps you can see, you see some flashes here occasionally. These are neurons where there's a calcium influx because they're spontaneously communicating. So they're thinking. They have nothing to think about. There's no input and no output, but at least they're trying to form circuits. And this is a brain functionality which helps us a lot. But they're also showing beautiful myelination. This is far too small to see here, but you might see this is oligodendrocytes which are wrapping themselves around the axons. After 8 weeks of development, 40% of the axons are myelinated. So really an interaction of different brain cell types producing cell functionalities. And this is opening up at the moment for demyelination, a toxic effect of some substances, but also multiple sclerosis research and others. As I said, we are interested in autism, so we are applying our model to these type of diseases. Our belief is that it is a combination of a genetic disposition and an exposure to toxicants which is giving us the effect. You need to have sensitive genes, and then the exposure to a chemical hits. That's our theory we want to show. So what we're doing is we're producing mini brains which are either coming from autistic backgrounds, that's on the list still, but what we just have been doing, we introduced with a partner, Herbert Lachmann from Einstein, he he did introduce risk genes of autism into healthy donor background, and we see some synergies between them so that we can observe such effects. So this is, I don't want to go into the science very much, but this was recognized already 2 years ago when we announced this, that this is an opportunity for replacing animal testing. This is the front page of the Financial Times worldwide, and we had mass-produced mini-brains to spark rethink over drug testing on animals. So there's a lot of expectation raised, and in the meantime, more and more people are also commercially producing them. We have established projects on stroke, on trauma, on toxicity. I think today or tomorrow, a paper on infecting these mini-brains with viruses, Zika, HIV, J.C. Cummings virus, dengue, will be published, where we also introduced immune cells, the microglia of the brain, which is invading the brain in embryonic development. We're interested in the neurodegenerative diseases. We have projects on Parkinson's, Alzheimer, amyotrophic lateral sclerosis. So you can start studying enormous things. So these are really enabling technologies that do much more than an animal experiment could offer. Here you see the mini brains. They're like small snowflakes, just visible, so that you can actually pipette and place them. But it's open up also for interesting new personalized medicine approaches. What you see here is glioblastoma. We grafted our healthy mini brains with cells from a patient glioblastoma, not this patient. I only put this up as a reminder that this is not only a childhood disease, but also has very prominent adult patients. This is, you see, the tumor in a natural environment. And now we can test either new drugs, and we show this in the first paper, which is at the moment under review, but you can also think of personalizing this, because each and every glioblastoma is different. We have 200 different chemotherapeutics. We might be able to choose the right one for a patient. And again, this is not replacing an animal experiment. There's no animal experiment which could do this, perhaps a newt mouse in which we graft some tumors, but that's not really practical. It is enabling something completely new. So that it is really the beginning of organoids, of the beginning of culturing cell culture, which has some architecture and some functionality of an organ. And this is what we believe, but it has to be shown, a more meaningful tool to make predictions. But we need to do this. But we also need to place this in a strategic development. Safety sciences has developed like a patchwork. And my mom did this patchwork of tox for me, you see here, because I want to illustrate. Each scandal gave us a patch. Thalidomide, reproductive toxicity testing, 2 generations. Eye irritation was the consequence of the lash lure scandal in the early 30s. So there's very often it is a response, a fix, adding something, while I believe that the new technologies will require that we are systematically building something up, that we are building a system based on biology, mechanism, what we understand with the tools of the 21st century. Because I'm now 55 years old. The tests we're using in toxicology were mainly defined when I was not yet born in kindergarten. I think it's about time to make some change. If the knowledge in the life sciences really doubles every 7 years, we have now about 1,000 times more knowledge than when we designed these tests on which we are still relying. So there must be something in the box, and I think we should do this strategically. This is more a problem in Europe than it is in the US, because here in the US we have, at least over the last few years, a wonderful collaboration between FDA, EPA, and the NIH as a TOX21 consortium, which I think is really a remarkable thing. Okay, I would like to tell you something more. We're also trying to get this message out. We're implementing this in curricula. We're trying to get this into more and more study programs here in Hopkins. I mentioned earlier that we have a humane experimental techniques at animals in law classes and certificate courses. We started last year with a lecture series on toxicology for the 21st century, 13 classes. and one on evidence-based toxicology, is trying to bring evidence-based medicine into toxicology, systematic reviews and these things. Katja Zawin, he is our director of the EBTC collaboration. And Lena has the lead for our education program, and I'm bringing this not that to inscribe here in Hopkins, but we have made them available for free, these courses. We have put them on Coursera, which is one of these mass open teaching platforms. And you can see we have in the meantime already more than 700 active learners for the Tox 21 course. The evidence-based toxicology course went live only on the 22nd of October. But here you can for free study the new technologies, evidence-based toxicology. There seems to be really quite some interest. If you want a certificate from Coursera, it is $49 per course. So it's very affordable and obviously worldwide available. So let me close with 2 slides. One is, I believe that we are not yet in a position to really abandon all animal testing. There's far too many reasons why we need animals, why they're prescribed in legislations which we will not be able to overcome in a short period of time. But less and less is possible with the new technologies. But the most important roadblock is the overestimation of the quality of the animal models. And this is why we need to get the message out, the message which is so strongly supported in this book, in the various chapters, about the lack of quality of the animal models and the need for a paradigm change. So this is a very important message. And the last one, a word of an economist, John Maynard Keynes, who said, the difficulty lies not in the new ideas, but in escaping from the old ones. We really need to find mechanisms, like the forum just presented, like the many activities we are having with stakeholders and also lay audiences, to move away from the old and open the door for embracing the new technologies. Thank you very much.
Thank you, Thomas. That was positive and in the end negative, but still more positive. Any questions from the audience?
Yeah, actually it's more of a comment, because I guess one of your first slides, you put it with that the current usage of lab animals is about 115 million per year for safety testing.
No,
and it
no,
triggered...
No, not for safety test. This is all animal use.
That was all animal use, okay.
Yeah, so
but it sort of triggered, I guess specifically the toxicology, that there was a little Remember Today book published in 1937 called 100 Million Guinea Pigs. And the 100 million guinea pigs in the title actually referred to the population of the United States at about that time, the mid-1930s. And it was basically an expose on products like lash lure, and saying how and it basically almost says how we got to where we are today with the animal testing, because prior to the passage of that new Food and Drug Cosmetic Act in 1937, products were put on the market, and if people died, then they were pulled off the market.
Yeah.
So I think that's one explanation. You wonder, how did we get to this point of all the animals? And it actually probably goes back to that book, that 100 million guinea pigs. So the 100 million people were actually replaced by 100 million animals.
I mean, to some extent, we have this situation. As I said, the untested chemicals, that is a mass experiment on the population. The numbers are a bit old. They're coming from Katie Taylor, who was supposed to be here and was so nicely substituted for. We did in our paper on the omics, most important omics is economics, we did actually try to validate whether the numbers are still correct, and checked for a few countries, and we have no reason to assume that they're completely off by now, 10 years later.
Any more questions?
I think everybody's longing for a chill-out glass of wine now, Friday evening, afternoon.
So, we only have the afterword left. That will be, thank you, Thomas. And then afterwards, if you want to join us, we will walk to the inner harbor and have food and drinks. So anybody who is up for a walk, please come and join us. So next up is John Gluck. I was lucky to meet him and honored to meet him last year, actually, because a friend of mine, Elizabeth Omendi, organized, she has the Animals in Science Policy Institute, and she organized a talk with John because of his memoirs that he wrote, and they were published in 2016. And they are called Voracious Science and Vulnerable Animals, a Primate Scientist's Ethical Journey. And it was a pleasure to meet you last year. And the book I actually reviewed for the Altex journal Tierethik, Animal Ethics. And it was very captivating. It was amazing. It was a good, like, I can highly recommend this book. You get an insight in how it is to do animal research, what the thought culture sometimes is, and yeah, that you still raise ethical questions, of course. So thank you, John. Thanks for doing the afterword, and thanks for giving us some final words for our book launch today. Thank you.
What a day. I've always tried to respect the caution in the Talmud that one should be very careful about holding forth in front of their teachers. And by the end of this day, I can say I have many more teachers than I started the day with, and I thank you for that. But it makes holding forth here, even for a few minutes, more difficult. So I'll take the recommendation of Plato and not try to teach ethics, but just to remind you of it. After I'd finished reading the book that we've heard about so deeply today, and had the opportunity to write an end note, what came across to me was, I think, what people have been talking about all day is, and it's like, what is in the way of change? What gets in the way? And we've heard, some very important leads to that as we've talked today. But of course, there is a large literature on why scientists stay where they are so frequently. There's a very large literature about the sociology of science. One of my favorite books is by Ludwig Fleck in 1935, where he coined, I think, what are very important concepts. And they were the concepts of thought collectives that scientific groups or areas or laboratories actually at times create what he called were thought collectives. That is, groups of people who thought the same way, and that they started to act like what happens in the street in groups. Resistance, making it hard for others to join, and throwing out people who didn't accept the norm. And he also talked about these thought collectives producing thought styles. And these thought styles, that is the way to think about science and the topics of interest to those scientists, can get so restricted that a person has a thought style and they don't know what you're talking about when you talk about some of this. It's like, it's not so much necessarily a complete rejection. They don't understand it. And so Fleck had a lot to say about how thought styles and thought collectives have to be managed, or how a laboratory has to be managed to prevent the creation of those very inhibiting structures. Of course, as we heard, Thomas Kuhn, talked in his magisterial work in the 60s and 70s about the power of paradigms to limit and to close and to resist change. And he called that normal science. Bruno Latour wrote a book in, I think it was 1979, where he was an anthropologist who went and spent 2 years in an office in Jonas Salk's laboratory, just kind of writing down what happened. How did facts get created? What was flowing in there in those laboratories? And he said, because you can't ask a scientist about what's going on, because the scientist will give you some ideal way that they imagine that they're working. And so instead, it's a tribute to Salk that he allowed this person to come into the laboratory and just sit there, writing about and looking about, and sometimes writing second to second, minute to minute about how people were interacting, but giving a picture, again, of the social dynamics of change or lack of it. So I think the last thing I want to say today is, or I want to interject today, and instead of going into the 21st and 22nd century like we just visited, I want to go back to the 19th century and tell you just for a moment a thought experiment that a philosopher, mathematician named William Kingdon Clifford poised for people to consider. And Clifford was interested in the ethics of belief. And his essay, which was published in 1877, entitled The Ethics of Belief, is, I think, pretty well known, and it's known well for at least one reason for sure, and that was the thought experiment he put out there for people to consider. And the thought experiment was he asked us to imagine or envision a ship owner. And the ship owner has a transatlantic business where he takes vacationers and business people across the Atlantic and deposits them in New York and then back in London. And he likes his business, he does well in it, but he's starting to have questions about the seaworthiness of his ship. And what he should do, whether he needs to stop and delay these trips or not. And he goes and examines his ship as closely as he can. And he comes to the conclusion that it is, and it's a sincere conclusion, that it's seaworthy. And so he starts selling tickets again, and one of his ships goes to the bottom in the middle of the Atlantic Ocean. And Clifford wants us to ask ourselves, well, was he responsible for those deaths? And he kind of puts it this way. He had no right to believe the evidence that was before him. That is, he may have, he had a sincere belief that what he was imagining in terms of the quality of his ship was sincere. But he was saying what Clifford wants us to say, but he didn't have a right to have that belief. He had acquired his belief not by honestly learning it in patient investigation, but by stifling his doubts. So he comes up with a principle that I think is worth thinking about, if not debating, given the lack of time. This principle is that it is wrong, and it is always wrong and everywhere wrong, for anyone to believe in anything on insufficient evidence. There's too much at stake. What this tells me is that that it's not just who's the what group's the winner or the loser or the individuals that feel success in their research, and it's also or can make a good argument. We've all met academics and colleagues who can argue just about anything, and we can get damn good at it. But the question is, do you believe it? And do you have a right to believe it? I guess what I'm trying to say here is that Clifford's assertion about the ethics of belief should continue to inform and haunt us. And that's those of us who are building alternatives and those of us who doubt them. Thank you.
Thank you. Are there any comments, questions for John?
They're tired.
Speechless. Okay, I just have some comments. If you want to see the table of contents, we have some copies here. We also have an email list laying out, so you can put your email and we will inform you when the book is finally available. And we also have, if you want to buy this expensive hard copy, we have a pre-order form where you get a discount. So just, I don't know, who wants. But the email lists are here, the table of contents. Why are you laughing?
It's not a very good salesperson.
I mean, we have all those great funders who gave us the money. I don't need to sell this book, but I think it's going to look amazing in hard copy. So if you want to have one, then yeah. But thank you everybody for having been here today. It was wonderful, and also, as I said briefly in the beginning, we will have a book launch in the UK on the 8th of March that is hosted by my co-editor Kimberly Jane. And some of our speakers will be there, and as well as others, like some of our today's speakers. And then we will have another one in Munich on the 14th and 15th of June. And we also plan something in the European Parliament in next fall after the elections to talk to the politicians there. So stay tuned. Thanks a lot for coming, and yeah, who wants to join us for, we will walk down to the inner harbor and have drinks and food. Please join us if you want to. Thanks a lot.